My lab is kind of more of a basic science lab, and we are interested in the cellular and molecular mechanisms of neurodevelopmental disorders. And as geneticists and cellular and molecular biologists, we rely on researchers like Jill to take what we find at the very small cellular level and go out to the organismal level. So a lot of the work we do, we try to go from genes to cells all the way through organisms and behavior. So my talk today, I'm going to avoid data for most of it. And this is a challenge for me. I love to go over the data, but I'm going to try to keep kind of high level and talk about three big swings in our understanding of neurodevelopmental disorders that I've been a part of and that my research is hoping to address. So there's three parts to my talk. First, I want to talk about this last going on near 20 years that has really transformed our understanding of the genetics of neurodevelopmental disorders. And I'm specifically focusing on moderate to severe disorders that often include comorbid autism spectrum disorder diagnosis, intellectual disability, as well as epilepsy and frequently dysmorphology. All this was lumped together for many, many years. In the last now 15 to 20 years has been a progress of separating out different specific cohorts or different specific monogenic forms of these disorders, and that's what I'm going to focus on today. So I'm going to talk about the progress we've made in the last 15 years or so in understanding the genetics of these severe neurodevelopmental disorders and how that informs basic researchers like myself and gives us kind of this treasure trove of information to start with to try to understand what we call the molecular mechanisms underlying disorders such as autism and intellectual disability. I'll talk a bit about our specific research area, which is trying to use basic science and models to understand the impact of patient mutations on brain development and brain function. And then at the end, I want to introduce a new area that is emerging now, and that's using our genetic knowledge, using our understanding of which gene might be mutated in any given individual or individual patient, and trying to develop molecular technologies that allow us to rescue that specific mutation or rescue expression of the gene. And I'll talk more about that at the end. And this is a kind of an opening area right now. So my journey in working on the genetics of neurodevelopmental disorders started when I was a PhD student. And this is in the kind of onset of what I think of as the genomic era. And let me give you a little background of where we are and how we got there. So monogenic neurodevelopmental disorders is a specific subset of individuals who are diagnosed with generally moderate to severe neurodevelopmental disorders. As I said, often exhibit comorbid autism spectrum behavior or kind of those behavioral phenotypes, as well as developmental delay or intellectual disability, and frequently epilepsy as well. Monogenic is a term geneticists use to refer to a situation when the phenotype or the disorder is caused by a single gene or by mutations that impact the function of a single gene. So monogenic disorders basically have a root in a single mutation in a single gene. There was some early success in mapping monogenic forms of neurodevelopmental disorders. Two of the best known and best researched monogenic forms are Fragile X disorder, which is caused by a very specific mutation in the FMR1 gene, and Rett syndrome, which is caused by a variety of different mutations that impact the MECP2 gene. So these two were the first single gene neurodevelopmental disorders identified. And what's similar is that individuals that have either the stereotypical mutation in FMR1 or the variety of mutations in MECP2, they present with a fairly consistent set of behavioral, cognitive, and dysmorphology and other clinical presentations. So we recognize these, or geneticists have recognized these specific forms, but up until we had access to individual patient genomes, we really had a blind spot for the other forms of monogenic disorders. So we only, as geneticists, could identify what we call the causal mutations, or the specific mutation that any given individual carried that led to the disorder, in a very small proportion of patients, and generally through these monogenic forms or through syndromic forms when there is characteristic dysmorphology also present. So this is a plot showing the diagnostic yield for intellectual disability. And I'm using moderate to severe intellectual disability as a representative phenotype, but this is true for all moderate to severe neurodevelopmental disorders. And the x-axis shows year from 1970 to 2015, and the y-axis shows diagnostic yield. And diagnostic yield simply means, can we find the causal mutation? So can we find a genetic explanation for, in this case, moderate to severe intellectual disability? And you notice that aside from this bump here, this line is largely flat and very low. And this is in this era when there was a lot of advance in other phenotypes, other complex human diseases, through approaches such as family studies, trying to find mutations that traveled in families. But those types of approaches didn't yield payoff, or we weren't able to identify many genes that were responsible for these complex neurodevelopmental disorders. In 2000, that's just after Rett syndrome was mapped to MECP2, which was mapped through a kind of painstaking process of going gene by gene. The human genome was released in 2001. And in the years following the release of the first human genome, or composite genome, we had access to new technologies that allowed us to look at individual genomes and search for mutations present in those genomes. And the impact of these technologies really had a transformative effect on the field. And this transformed both translational and clinical approaches, as well as basic science research. So what you see in this part of the plot is this inflection point when various technologies were kind of arose that allowed geneticists to start looking at individual patient genomes and look for causal mutations. Initially, we were able to detect very large mutations. And in my PhD, I worked on some of these projects, writing computer software to try to identify these mutations in populations of cases and controls. I made very small contributions in my PhD to this kind of this rise in our understanding. And later on, as I started to kind of launch my own career, I decided that I wanted to focus on understanding what the impact of these mutations were. But let's coming back to this approach that allowed us to map these mutations and understand which genes were associated with intellectual disability and other neurodevelopmental disorders. This inflection point here is really paralleling the inflection point of the reduction in the cost to sequence the human genome. And what happened around the same time here is we gained the technology to rapidly and cost effectively sequence or screen or scan an individual's genome to look for mutations that might be causing a phenotype such as neurodevelopmental disorders and might be present, for example, only in a single case. And this is really what has changed the diagnosis of intellectual disability and other neurodevelopmental disorders. So it's not just any mutation that geneticists have looked for that really led the change. It's specific types of mutations that have been so important. So from the decades of work on the genetics of intellectual disability and neurodevelopmental disorders, we knew, or we assumed, that the majority of patients were unlikely to share mutations in the same gene. And similarly, that if there are mutations that were causing these disorders, they were unlikely to be transmitted from parents in a large proportion of the time. And that assumption was because in general, cases of intellectual disability and severe neurodevelopmental disorders are not familial or don't travel in families. And there are some exceptions, of course. So geneticists started this process by looking for specific types of mutations. First, the mutation is present in the case, but not the parents. And we call this a Denovo mutation. I'll talk about this in a minute. And then with the presumption that these mutations had a very strong effect we could exclude mutations that were present in undiagnosed or healthy controls as being unlikely to lead to these phenotypes. And so to give a sense of what this looks like, this is a very simplified schematic of searching for mutations. But if we have a case and we have the DNA from the patient or the case as well as the biological parents, we can find which mutations or which sequence changes, so changes to the DNA bases, are present and whether they were inherited from one of the parents or not. So here in white in the case are all the bases that are non-changing. So humans in general share most of our bases. There are some estimates of around 1 in 1,000 bases will be different between two individuals in our genome. And here, what is shown are individual base changes or sequence variants that are transmitted or inherited from the parents. So here's a G that was inherited from the paternal, the father, and a C inherited from the mother. So the G is not present in the mother, and similarly, this G is not present in the father. But we can map these changes in the child to the parent's DNA as well. What's shown in yellow here is one of these special mutations called a Denovo mutation, where neither parent carries that sequence or that sequence variant or that mutation. It's present only in the child. De novo mutations arise either during germ cell formation, the production of sperms and egg, or early on in embryonic development in one of the original cells in the embryo. And we know now through much work that these de novo mutations seem to be biased towards occurring during spermatogenesis or in the formation of sperm. Not only were geneticists looking for these de novo mutations, this very special class of mutations, but they were also looking for mutations that had a predictable impact on the function of genes. So I won't go back to high school or college biology, but there's this central dogma of molecular biology where our DNA is first transcribed into an intermediate called RNA, which is then read out and translated into protein. And so proteins are the building blocks of a cell that actually perform the functions in the cell. So genes, in the end, produce proteins, and the proteins are used by the cell. And so geneticists looked for mutations where the mutation was first de novo, and second, would impact the production of a protein such that the mutation would cause no functional protein to be produced. So in looking for this very special class of mutations that are de novo or only in the child and have a major impact on the production of the protein, geneticists were able to identify the causal mutations in a large proportion of moderate to severe intellectual disability cases. And what this ability to look at individual genomes did was really change how we think of the genetics of these disorders. So now if a patient is referred with idiopathic neurodevelopmental disorder or intellectual disability, simply by doing a genome screening test, a diagnosis can be achieved at the molecular level around 50 to 75% of the time. And what we learned from this, looking across many cases or large cohorts of cases, is that we can think of the genetics or the genetic causes of these disorders as not a single disorder, but instead a collection of rare monogenic disorders. And so what the colors here represent are, in each patient or in each case, there may be a different gene that's mutated. And there are a small number of patients that carry mutations in the same gene for many genes. So the impact this has had is really hard to overstate, I think, from an initial genetics perspective and a genetic diagnosis perspective. Geneticists refer to this kind of structure as they have genetic heterogeneity. And that means that there are many different genes that are implicated in causing severe neurodevelopmental disorders. In each gene, any two cases that have mutations will frequently have different mutations in that gene. So most mutations themselves are specific to a single case. But by leveraging the information that we get by looking at many hundreds, or now at this point, many thousands of cases, we can build up evidence of which mutations might be causal based on the total number of aggregated mutations in a gene exhibited across cases. So we can now think of kind of neurodevelopmental disorders as a collection of monogenic or single gene disorders. And this is true. The estimates vary, but some common estimates are 10 to 20% of autism spectrum diagnosis, and as I said, 50 to 75% of intellectual disability at the moderate to severe level. So especially where you have comorbidity between these phenotypes and the impacts are fairly severe, there is a high probability that these can be assigned to one of these kind of newly understood monogenic forms of neurodevelopmental disorders. So previously, these patients would come to clinics and be diagnosed based on some aspects of symptoms, often at the behavioral level, or for some known dysmorphology that would allow us to identify a syndrome. Now, with this kind of transformative impact, genome sequencing, or at least sequencing parts of the genome, has become a first-tier method to obtain a diagnosis in severe neurodevelopmental disorders. And indeed, in many severe, what are presumed to be genetic disorders, this is true. The Mayan Institute and UC Davis Health System has, it's not new anymore, but a relatively new division of genomic medicine that accept referrals for patients that are likely to carry a mutation, and the specialists will then review the case and decide whether it is worth performing genome sequencing. But by having access to genome sequencing, this has prevented what are kind of referred to as the diagnostic odyssey that some of these patients or cases had to go through before, where now we can look for the causal mutation up front and then ask questions about the care and treatment later. So while this change is still evolving, we now can kind of identify causal mutations, but we're at the early days where there's generally not specific treatments available. Nonetheless, just having a genetic diagnosis can really have a big impact on patients and families, for clinical care, for preventative care, but also for support systems where now individual patients that carry mutations in the same genes can are forming support networks, often have similar clinical presentations or similar challenges. So this has really transformed from a clinical and translational perspective our understanding of these disorders. And I say that as a basic scientist, as a human geneticist, to introduce this, but this is an area that it can be much better talked about by specialists, by the clinicians that are involved in this process. From my perspective, as a basic scientist, this genetic change has been a watershed moment similarly, because it gives us new entry points to try to understand the underlying biological mechanisms of severe neurodevelopmental disorders. So that's the end of the first part of my talk, where I wanted to introduce this idea of changing our perspective on neurodevelopmental disorders to an understanding that they're a collection of rare monogenic disorders that doesn't account for all patients, but accounts for a fairly large proportion. And then transition to what do we do next? Now that we have a genetic diagnosis, there is changes in treatment and care, but there's also a starting point for us to design studies to understand why. Why does a mutation impact brain development such that it results in these behavioral changes or these cognitive changes? So the most recent study now was looking at around 12,000. This is specific for autism spectrum disorder cases, where we can now compare the genomes across 12,000 cases and many more presumed healthy controls to identify the mutations and then the genes that are associated with autism spectrum disorder at the monogenic level. So this is a recent study that was published last year, but has been floating around for a few years, where the list of genes has now risen to over 100 genes are now known as causing monogenic forms of neurodevelopmental disorders, and specifically overlapping with ASD or autism diagnosis. The plot that I just pulled up here shows the position of each gene in the genome across the chromosomes on the x-axis, and the level of certainty or confidence in that gene being associated with autism spectrum disorder based on the number of patients that have mutations in that gene. The little red dotted line is the level of statistical certainty or significance that's currently used as a threshold for saying that a gene is associated with autism spectrum disorder or can cause a monogenic form of the disorder. And so there are 102 genes that are above this red line, but you can see there are some genes that are much higher, and these are ones that have risen to the forefront of our study and are the focus of much basic science research now. These genes at the top that are listed with gene names are a variety of different types of genes, but are kind of the first tier that have been looked at or are being researched by basic scientists like myself. So I want to talk a little bit about how we go from these gene lists to an understanding of biology. Okay, so even just from these list of 102 genes, there's already many surprises and many questions to be answered. There's also huge challenges. So with over 100 neurodevelopmental disorder or autism spectrum disorder associated genes, where do we start? How do we start to digest this list? In addition, many of the genes that come up, very little is known about these genes. So while some are well studied, such as MECP2 or FMR1 in Rett syndrome and Fragile X, many of these newly studied genes, there hasn't been a model, haven't been studied exhaustively, so we know relatively little. Next, what might a mutation impact? So the brain is an exceptionally complex organ with many different cell types, both many different types of neurons and then non-neuronal cells that support neurons, such as oligodendrocytes, astrocytes, and microglia. Which cells might be impacted by these mutations? As these severe neurodevelopmental disorders are fairly early onset, the current model is there must be some alteration during brain development. But even so, that's a span of years that we have to think about for when the specific impacts might be occurring. So how do we start? What do we start with? So the excitement from scientists like myself is that while this is an exceptionally complex problem, there's some really interesting kind of signposts to start with. And so one of the ideas has been looking for what we call convergence in what these genes do. And convergence means where do they overlap? Where are their shared functions? So I'm going to go into that a little and talk a little bit about what my lab is doing in this area. So from this publication, it confirmed what now is around a decade of research on these autism-associated genes. One of the first questions that has been asked is, are there autism-specific genes? Or are there just general genes that are associated with autism and intellectual disability or these severe neurodevelopmental disorders? And from this kind of most recent update study, the answer to me is pretty clear that most of these genes that lead to monogenic forms of neurodevelopmental disorders contribute to both cases diagnosed with autism and with intellectual disability, meaning there's a strong overlap in these genes. And this plot here shows this, where the authors separated these 102 genes into genes that are increased burden in autism-specific diagnosis versus both present at similar levels or increased in intellectual disability or other neurodevelopmental delay. And while there's some separation of these two classes of ASD predominant genes or ASD plus intellectual disability or neurodevelopmental delay, they largely overlap. And every gene where there are multiple patients, there is some patients show comorbid or exhibit both of these diagnoses. So the answer there is pretty much no. We're at a point when there's been more or less a dead end when we talk about these severe presentations of autism-specific genes. My lab works on one gene that I'll talk a little bit that there was a lot of hope would answer questions specific to autism, and that's the gene CHD8, which has a higher burden in autism spectrum disorder patients. But even there, the comorbidity is so strong that we really are now thinking about these as brain genes rather than autism genes. The second and kind of root point for my lab and what we're looking at is, what are these genes doing? Can what these genes do teach us something about the underlying biology of autism and intellectual disability? And surprisingly, and this has held true now over a decade, there have been two main classes or two different types of genes that are mutated in cases. The first is this surprising class of genes that are involved in a general cellular process called gene expression regulation. And I'm going to talk about that a lot more in a minute, so I'll come back to that. The second and more expected finding is that many of these genes are involved in neuronal communication. So they converge in function at the synapse, or in the function, the generation or function of synapses or neuronal circuits. So there's two main types of genes here: the gene expression regulation, which are the surprising class that I'll talk about, and then the more expected genes involved in neuronal communication or synaptic structure. The final point of convergence is in which cells in the brain matter for these disorders. And to look at this, researchers look at where are these genes turned on in which cells. And the overwhelming answer for the monogenic forms of neurodevelopmental disorders is that these genes seem to be essential in neurons. And so there's some question about whether it would be neurons or non-neuronal cells, such as the neuroimmune microglia, that may be impacted. By and large, the current model, based on the findings that we have now, is that the majority of these genes probably impact neuronal function or cause neuronal dysfunction underlying these disorders. So my lab, and what I was interested in, is what about this special class of gene expression regulation genes? What makes these genes, when mutated, cause autism and intellectual disability? And one further point here is these gene expression regulation genes, shown in purple in this plot here, they have their highest expression, or they're turned on at the highest levels in the prenatal period or during brain development. And this suggests that there is this point of convergence where mutations to these genes impact early brain development through altering gene expression regulation. So gene expression regulation is this general process. So each cell in our body, so it's an old illustration showing the skeletal, muscular, and organ systems. Each of these cells that are very distinct has the same genetic instruction manual, or human genome. And that instruction manual contains somewhere between 20 and 30,000 genes, depending on how we define a gene. But think about it as each gene makes a specific protein that's needed. And in any given cell, for example, neurons in the brain or cardiomyocytes in the heart, those cells only need some subset of the total number of genes in their function. And so cells in our body have the ability to regulate the expression of a gene, and expression means the production of protein from the gene. And so gene expression regulation is the process by which cells are able to turn on or turn off the specific genes in order to generate only the proteins that are needed in that cell. So this is important for, the overall, like, large-scale differences in skin cells, heart cells, brain cells, but also within cells in the brain, within neurons. Neurons actually have very different morphologies and functions. And the ability of neurons to take on these specific shapes and structures and functions requires very precise control of the production of proteins, or very precise control of gene expression regulation. And so this suggests that in severe neurodevelopmental disorders, the mutations in these genes that are important in gene expression regulation are causing a loss of neurons to have this tight control over which proteins are produced when. So this gives us a basic model to think about. Now we want to know, well, what is the impact of this, or is this model correct? So my lab is both interested in the basic science questions of how is our brain encoded in our genome and how does gene expression work, as well as in questions of how do the genetic and environmental causes of neurodevelopmental disorders alter the brain. So we think about ways to model these disorders, generally using mouse models, though we do some non-mouse modeling as well. And we use mice because brain development in mice undergoes very similar processes to brain development in humans. And so we can model the changes in developing brain that occur in the presence of one of these mutations. So our approach has been to start working through the genes that are associated with gene expression regulation and ask, what are they doing during brain development, and what are the impacts of mutation on the brain from the level of cells all the way through behavior. So I circled 3 of the genes that my lab has worked on. We have a major program working on the gene CHD8. We've also contributed to work that Jill has led on ARID1B, and have worked on a few other genes, including a gene called POGZ. These are all the ones that are kind of published or currently close to publication. We've worked on some of these other regulators as well. And the reason that we're kind of spread our effort across these genes is because we expect that they are going to converge or do similar things in the developing brain. So the genes that are circled in red are the genes that are at the highest level of certainty or highest level of statistical significance for being associated with autism, where we know the role of the proteins produced by these genes is in turning on and turning off genes during brain development. So for example, we use mouse models where we can engineer the mice to carry a mutation similar to a patient mutation. And then we can study across dimensions what changes in the brain. For example, we look at neuroanatomy, brain structure, and behavior through collaborations, for example, with Jill Silverman, David Amaral, Jackie Crawley here, and then other researchers elsewhere. And then we use what's known as genomic approaches to look at the molecular and cellular changes in the brain across development. So we're really trying to understand kind of when there is a mutation present and we use a mouse model, what changes across this whole window. And so we work across different models towards the goal of understanding each of these individual monogenic disorders at depth, but also towards understanding similarities between them that might give us more broader understanding of these disorders. So, you know, we've done a lot of work in this, and our lab is one of many labs that is doing this research. I instead of going into depth of where we are, I think it's fair to summarize that we've only had these gene lists for, you know, at most 10 years now, and this is still fairly early in understanding why these mutations cause disorders, but we've learned a lot already today. So I just want to return to this diagram, which shows all the different genes that are associated. So at this point, 102 different genes, and somewhere around half of them seem to be playing important roles in turning on or off genes during brain development, turning on and off gene regulation. So we've studied a number of these, and all the genes that we've looked at, when we make the mutation or representative mutation that's similar to what's found in patients, there's an impact where the precision in the production of proteins is altered. That is the cellular impact. That then causes alterations to neuron structure, function, and development. So brain development is this sequential steps of building a brain from early stem cells, which expand during very early embryonic development, to the birth of neurons, which then migrate to form the structures in the brain, and eventually to the formation of synapses. And across each of these steps, we've seen alterations to brain development in mutant mouse models that we're using to study these genes. These changes during development and within neurons then lead to impact at the circuit level and at the behavior level, and that's why we work with neuroscientists and with behavioral scientists to understand what the root cause of the loss of gene expression does when we take it all the way out to the behavioral models. So as I said, we're still in pretty early days of understanding what each of these monogenic disorders, what the impacts are, but we found some exciting convergence already in that many of the same genes that are needed during brain development are, we say, they're dysregulated, or the tight control of gene expression is lost in each of these mouse models. So we need basic science to understand the neurobiology of severe neurodevelopmental disorders. And by having these gene lists to start with and to ask what's similar across genes, there's great hope that in the next 5 to 10 years, we'll start to have these more generally understood models of what the underlying neurobiology of these disorders are. So that is the end of the second part of my talk, where I wanted to kind of show how, now that we have these gene lists that are associated with autism spectrum disorder, but also generally associated with intellectual disability and overall neurodevelopmental disorders. How having these gene lists gives basic scientists targets to start with towards understanding or building these models to better understand these disorders. And again, I'm not going as far into the findings because it's exciting. There's a lot coming out all the time on this. And if you look at some of the kind of complex human disorders where we've known the genes for decades now, we still are learning. So I anticipate that we're still kind of in the early stages of building these models based on our understanding of what happens after these mutations. So for the last part of my talk, I wanted to switch over to emerging an exciting area that we're involved in and that we work with other Mind Institute investigators on. And that's leveraging this understanding that these mutations in specific genes lead to these disorders to try to develop precision medicine treatments based on rescuing expression of the genes that are mutated. So neurodevelopmental disorders, the monogenic forms that I've been talking about today, are largely what's known as caused by haploinsufficiency. So humans have 2 copies of each gene. We have 2 copies of each chromosome, one inherited from our mothers, one from the father. And these 2 copies carry generally identical copies of the genes aside from specific mutations. In these monogenic neurodevelopmental disorders, such as ones in this plot that I've showed a few times now, one single mutation in one of the 2 copies of these genes is generally mutated. As I said, these de novo mutations result in a loss of protein production or protein function from that, what we call allele. But in this case, there's generally the second functioning copy of the allele still producing protein. But in haploinsufficiency, that one functioning copy isn't enough. So one copy of a gene is not sufficient for function. As an example, with our work in the CHD8 mouse model, or looking at single mutations to CHD8, what's shown here is the amount of CHD8 protein produced in wild types and a heterozygous or a mutant animal that has one copy mutated. And you can see that the gray bar is about half of the protein is produced in these mutant animals. Just reducing the amount of CHD8 protein produced in neurons or produced in the brain by half leads to profound effects, leading to brain overgrowth, neuronal dysfunction, and ultimately behavioral changes that our lab and other labs have found in these mutant CHD8 mice. So this is an example, like I said, of haploinsufficiency, where one of the 2 copies of the gene is broken, essentially. And this raises a question, well, can we, instead of, for example, coming up with a drug that selectively targets some aspect of synaptic physiology, can we treat the root cause by using the remaining functional copy of the gene as its own kind of internal drug itself and turning up the expression or turning up the amount of protein produced from the functional copy to return it to the amount that's normally produced in the neuron? So this idea is that we can rescue haploinsufficiency. Now that we know which gene is mutated, we have a target. How can we turn up the functional copy of the gene that still remains in all of our cells to get back to the normal amounts that are made? So I just want to switch over to a different gene that we work on and a different disease model we work on. So mutations in SCN1A cause a severe neurodevelopmental disorder known as Drave syndrome. And SCN1A is a gene that produces a sodium channel that's necessary for synapses to function. So my lab has worked on understanding the regulation or how the SCN1A gene is turned on in neurons so that sufficient production of the protein, which is called NAV1.1, is present. So this is a normal mouse showing a sagittal section of the brain. And the green color here is a stain for the production of this protein NAV1.1 that's made by the SCN1A gene. In our mutant mouse, we have a mutation that reduces the amount of protein produced from one of the 2 copies of SCN1A. This is the situation of haploinsufficiency. And you can see that there's a reduction in the amount of green, and that's a reduction in the amount of protein. But there's still protein there because there's still one intact copy of the gene making its full amount. So we've developed this new mouse model and recently are kind of in the process of publishing it in the next few days, hopefully. And at the end of it, the next step that we wanted to do, thinking about this model, was to ask the question of if we can rescue some of the mutant mouse phenotypes, and specifically the seizure phenotypes here, where we have a electrophysiology or a seizure to go on rather than targeting behavior at the start. If we simply turn up the amount of SCN1A or NAV1.1 that's produced off the healthy copy of the gene, can we rescue these phenotypes to test this idea? So this idea that we can rescue haploinsufficiency is really enabled by major technological breakthroughs in the past couple of years that enable us to rationally design molecules that allow us to target one specific gene in the genome. And you may have heard of this technology, the Cas9 CRISPR technology. It's kind of the leading push in this. And what this allows us to do is bring this specific protein to a specific site in the human genome and rationally design proteins like this such that we can use them. We refer to these as artificial transcription factors, is one way to refer to them, where instead of editing or altering the DNA sequence there, we're altering how much the gene is turned on or turned off. So our idea was that we would use this approach to bring one of these rationally designed molecules to the SCN1A gene, thus turning up expression of that. And we did this in a proof of principle experiment in a human cell line, where we brought one of these artificial transcription factors to SCN1A, to the start of the gene where it's turned on, and we're able to increase the amount of expression off that gene. We were super excited. We were thinking this is going to be a great next step, and we managed to secure a modest grant to do this. But like all great ideas in science, we weren't the only ones who had this idea. And in fact, there are many different labs and companies now that are pursuing this idea. And our funder, which was a smallish foundation, called us back a month later and said, you know, we love your idea, but we think that the contribution that we could make to this project probably isn't the best use of our foundation money. And then about a month later, this kind of press announcement came out with one of many now companies, but this one had recently in fundraising managed to raise $104 million to pursue the same idea in the same disease model of Drave syndrome. So while disappointing to us because we were excited about performing this work ourselves, I think this is actually exceptionally exciting for the field because it shows the viability of this idea. And there are now a number of academic and industry efforts trying to use the same model across some of these haploinsufficiency disorders. So the idea here is that we can rationally design one of these artificial transcription factors to induce or increase the amount of protein produced by the functioning copy of the remaining gene. So there are many human disorders, genetic disorders, that are characterized by haploinsufficiency, not only in the brain, but across other systems such as metabolism, in blood disorders, a variety of basically complex human diseases. So this is a general idea and has been started to be developed. In the brain, there are some real challenges to doing this because of delivery and access and the precision of expression that we need to hit. So mutations in SCN1A and Drave syndrome might have been a kind of a first target for many groups working on this because of how well documented the mutations are and how well understood the phenotypes are. But we're kind of in the very early days still, where there's still a lot of question of how do we do this in a physiologically relevant manner. So there's questions of delivery and dosage of this kind of a therapeutic treatment effect. Our cells in the body have thousands of genes that are involved in gene expression regulation. The current first generation artificial transcription factors kind of are just turning on the dosage, trying to get it raised, but it's likely that we need a precision regulation to really get effective response. So we shifted our focus at that point and shifted our focus to trying to target this idea that we need precision. If we're going to rescue haploinsufficiency, we need to rescue it by increasing protein production to the right amount in the right cells. And so we're using a viral strategy to do this by leveraging elements in our own genome that are important for turning on genes to use to turn on that same gene in the right amount. So this is just, I wanted to show this just as a new area that we're doing. So this is unpublished work, but we're using viruses that can infect neurons to deliver DNA to raise expression of a target gene. So this is a proof of principle experiment where we have a red delivery control to test how well we're getting our virus into neurons. And then we have a green fluorescent protein that's our target that we're trying to induce. And you can see that we're able to, we inject the virus into the brain. The neurons are infected by this virus and then express this protein. And we chose, I know it sounds like a lot to inject the virus that then go into the cells and produce a protein, but actually this is how a lot of the gene therapy, the promising gene therapy approaches are working right now. So our goal is to utilize an approach that's already kind of in this clinical translational world with the gene therapy, but do it smarter by controlling the rescue at the levels that are the most physiologically beneficial. And this again is just showing that we can, using this approach of delivering a virus, cause cells to produce a specific amount of protein. And so you can see the green protein here. And this is just a proof of principle in a mouse brain. One of the areas that we're also interested is combining kind of our mouse modeling work with taking advantage of this new system of cultured organoids that are based on human cells. And so these are also known as mini brains or brains in a dish. So in this image here from a recent publication, these little spheres at the bottom there are spheres of early representative embryonic cells that go on to form the structures in the forebrain or the cortex. So there's a limit to what can be used with this kind of a model, as you can expect. We can't grow a brain in a dish completely, but these mini brains have enough self-organization that they actually can recapitulate some of the early stages of brain development. And if you recall, the earlier stages are the kind that are most perturbed by these gene expression regulation mutations. So this is work that we did in collaboration with the UC Davis Center for Regenerative Cures that have some expertise in this. Is we generated these mini brains and then tried to introduce our viral delivery and induce expression in here. So we're hoping we can use parallel in vitro or cultured models for human brains in parallel with our mouse models to do this work. So where are we with taking this concept and getting it into a clinic? So there is a huge amount of excitement because there are many monogenic human disorders that are associated with haploinsufficiency, including almost all the mutations that have been mapped through this process of mapping Denovo mutations in neurodevelopmental disorders work through or are presumed to impact the cells because one copy of the gene is lost. So almost all of those genes from that plot I've shown a few times are candidates for this. So there's a huge amount of excitement that now that we know the genes, and now that we're developing these new technologies to allow us to turn on expression of a specific gene, we might be able to leverage this. But there are also a huge number of challenges. So first is the one I raised, is how do we turn the gene on only in the cells that need it and only in the amount that's necessary? As an example, many of these genes that are linked to autism spectrum disorder and intellectual disability are also linked to cancer. So if we turn those genes up too much, we may be doing just as much damage as good. So we really need to figure out how to physiologically rescue, not just turn on it and blast it out. So that's an area that my lab is very interested in working in. Other groups, including groups at the Mind Institute and at UC Davis, are trying to address the issues of how do we engineer these and deliver these therapies. So the current approaches probably need a fair amount of refinement for delivery and use in brain. This is true of more general gene therapy approaches overall, is that the brain is a relatively challenging tissue type or organ to treat with this kind of approach. And we need to develop the kind of delivery methods that, for example, won't raise an immune response, will be effective across many different neurons or infect enough neurons to have an impact on the overall function. And then the final question, this is another area that my lab is interested in, is will this matter? And the reason we ask this question is we don't know what the critical period for when these mutations disrupt brain development or brain function is. And often we're not going to be able to treat until well after for example, the embryonic or prenatal brain development has completed. So what happens if we turn back on expression of some of these genes at a point beyond which, for example, early brain formation is complete? Will that impact behavioral phenotypes? Will it impact cognitive phenotypes? Will it impact seizure phenotypes or some of the other kind of now appreciated endophenotypes or specific aspects of these monogenic disorders, for example, sleep or other parts that are more specific than the overall cognitive or behavioral changes. To answer that question, we're starting with mouse models and asking, well, if we turn back up expression, which phenotypes are altered? And it may be that mice are good models for this. It may be that we need better models. We're now walking down a path where some of the kind of earliest monogenic disorders, Fragile X and Rett syndrome, have already been down some of these paths with some of these questions. So I think that we should be very, very excited about this new idea, but also really have to deal with some of these tough questions that's going to require both basic science and translational research to address. But I again wanted to highlight that UC Davis and the Mind Institute is an awesome place for this intersection of basic science with translational and clinical relevance. So my group is involved in some new efforts to use this kind of haploinsufficiency rescue in specific monogenic disorders, and we're hopeful that kind of through the kind of teamwork that we can do here, we can make some real progress in the next few years in understanding how to best do this and what can be rescued or what can we change if we turn back up expression. So that's the end of my talk, or what I was planning on kind of presenting today. Again, I tried to kind of tie these together with the uniting ideas that once we know the genetic causes of these disorders, we can then start to understand or study the underlying mechanisms, but we also might have new avenues towards treatment through rescuing the specific genetic insults. There's a lot to be done. A lot of exciting research will be coming out. Identifying these genes is just the first step in knowing kind of why do mutations cause these disorders, and in using this genetic diagnosis to actually shape treatment beyond what we can do currently. So with that, thank you for attending this virtual talk, and hoping for some great discussion after. Last slide, I showed many of these people who are doing the work in my lab through the talk, but want to thank the graduate students and postdocs and technicians in my group that have led this work, and also want to thank and acknowledge my many collaborators. Jill Silverman, who's introduced me as moderating, is one of our core collaborators here, but we also collaborate with many other UCD investigators as well as investigators outward. Genetics is a team science field, and without these collaborations, we wouldn't be able to make the difference. And then finally, to thank my funding sources that have made this possible, including some early funding from the Mind Institute and some support for postdoc in my group from the Autism Research Training Program. Both of these were really instrumental into some of the early successes that our lab had in these areas.
So there is a few questions that we have in the chat. When you refer to ID, I suppose ID meaning intellectual disability with no known etiology. Is this ID secondary to another NDD, such as fetal alcohol or Fragile X, or is or T21?
Yeah, so we kind of try to. Again, I'm a basic scientist here, so I should be very clear that I'm speaking from a basic scientist perspective of what's going on in the clinic. But usually, for the patients that are included in these studies, as much as possible has been excluded already. So these are individuals that have been excluded for Down syndrome or trisomy 21, have been excluded from kind of severe environmental insults or impacts, such as in the case of kind of known fetal alcohol syndrome exposure or dysmorphology, and are generally excluded based on known syndromes that have been recognized based on characteristic presentation. So these are kind of the ideas that of the the patients or the cases that have gone into these genetic studies is the goal is to take as many unanswered or unanswerable ones and look at those first. There's a second question that says, if you start with a known population that, for example, is susceptible to, for example, Fragile X or Down syndrome, and then ask for secondary genetic background hits, that's a second type of approach. But generally, these are the kind of, you can think about these patients as the ones that other things have been ruled out.
Let me get to the other audience questions here. So Ted Toll asks, is the prevalence of haploinsufficiency in neurodevelopmental disorder an indication that relatively benign genetic variations in gene regulatory regions might cause changes in gene expression levels, which could then cause slight changes in brain development? Could this be one explanation for such a wide variation in human brain characteristics such as personality?
That's a super awesome question and a very specific question. So I'll try to not go down too deep down that hole. So my lab and my interest, we spend a lot of our work trying to understand how regions of the genome that don't encode proteins work. And the short answer is yes, that's one of the models we're pursuing, that mutations outside of genes might have impacts on the expression of those genes also. And so that's kind of an active area. There's also the question of the severe forms might involve a gene mutation, but there may be some intersection with these less severe forms. And there's been some indication, I think the paper I showed a few slides from shows that there's some overlap with common variants that are linked to educational attainment and psychiatric disorders, but that it's still very early to make those links in autism and intellectual disability.
I am interested in the commonalities across ASD disorders, common mechanisms such as mitochondrial problems and GABA deficits or glutamate upregulation, so that a treatment for one of these problems could help multiple disorders. What are your thoughts on these commonalities?
That's again a great question, Rondi, on the question of what's overlapping. And so this goes into this idea of convergence that I brought up, where we don't need to necessarily understand every single change that's present, but if there's convergence in some of these specific changes, like the ones you mentioned with, GABAergic or glutamatergic neuron function, are two specific neuron subpopulations. If we can find, for example, that there's convergence across these monogenic disorders in those areas, it may lead us to insights on causes and treatments. So for example, if we have a pharmacological intervention or a drug that can specifically alter GABAergic or glutamatergic signaling, we can use the findings from these basic science studies to prioritize which monogenic forms may be most amenable to that treatment. So this is kind of like the big idea of precision medicine, right? Is that once we understand these molecular causes, we can treat the molecular causes. And it's a good point that we don't necessarily have to go all the way down to treat the root haploinsufficiency itself. There may be effective treatments that are useful across disorders based on which neurons are impacted or which signaling or circuits are impacted. Yeah, I think it's super exciting. Again, I wish, so in Fragile X, where the mutation has been known about for so much longer, that single kind of initial monogenic disorder is 10 years, 15 years ahead of these other ones for us to really have reliable info. But the hope is that we'll get there by groups that are taking approaches like mine to dissect each of these single genes to build up this understanding.
I wrote a little note, but I'm not sure if it hits, Rondi, that we actually have a grant that we're trying to put together with the title being Convergent Mechanisms. Just takes a little time.
And I want to, just to say one more word on that. Yeah, I think, we can dissect the heterogeneity of these disorders at the genetic level, but also at the kind of downstream cellular molecular circuit level as well. And that's where the convergence at that level may allow us to specifically treat some endophenotypes or some aspects of the disorders effectively. That's a hope, is that we don't, we're not searching for the silver bullet for everything, but this can give us insights on how to treat specific aspects.
Randall Riley asks, does the specific pair difference, the SNP, make a difference in phenotype or in is it just the gene that it matters in?
Yeah, super good question. So this is the geneticists, we're just now understanding the scope of the different mutations that are present in individuals. And I heard a great talk a couple days ago where one of these genes that was at the top of the list there is called SCN2A. And through workup, some groups at UCSF that we are kind of in tight with, showed that specific mutations can cause a bias towards epilepsy, early onset epilepsy versus autism and developmental delay. And it matters which mutation. The mutation matters if it changes the protein that's produced. And so there's two different classes of mutations that we think about. One is these mutations that largely ablate the amount of protein. So no protein is produced at all. All the mutations have the same molecular endpoint with no protein produced. Other mutations alter the protein that is produced in specific ways. And so one goal now is to take these 12,000 and growing cases where we now have many individual examples of mutations in genes and see if we can understand if there's specific behavioral or epileptic or cognitive outcomes associated with specific types of what we call alleles or mutations. And so, in the precision medicine era, eventually we hope each mutation will have enough information to treat that mutation specifically. But yes, the short answer is it does matter what the mutation is.
Polygenic risk scores have become increasingly popular over the past few years for identifying individuals with higher de novo genetic quote unquote burden for various conditions. What is your take on polygenic risk scores and the use for polygenic risk scores across diagnoses? For example, using a polygenic risk score for ADHD to identify cases with genetic overlap of autism.
That's an awesome, that's an excellent question.
Yeah.
Let me clarify a little bit about genetics here. So the area of my research in the talk today is focused on what we call monogenic forms of these disorders. And these are distinct from a different model, which is the overall burden of many mutations. And for those of you, who aren't familiar with this area, in some patients, the ones that I'm talking about today, a single mutation is assumed to be largely causal, although there's some variability that probably depends on genetic background still. In other patients, and especially in other disorders such as psychiatric illness, bipolar disorder, schizophrenia, major depressive disorder, etc., the genetic burden is assumed to be less due to single causal mutations and instead due to the overall contribution of many different alleles or DNA variants. And so we can sum the impact of all those genetic background in these polygenic risk scores. So it's kind of a different, we think about this as a genetic architecture where the level of complexity is much higher when it's many, many different, we think of smaller effect mutations contributing. And so there's an idea that these polygenic risk scores are going to be very useful for summarizing genetic background overall. And they're really distinct and separate from this idea of single causal mutations. And so that the area that the kind of has been most accessible to geneticists and basic scientists has been trying to understand what is the impact of these single causal mutations, because we really have one gene to pay attention to in a model so we can model it effectively. The polygenic risk score brings in genetic background overall and has been useful from the level of genetic epidemiology so far in understanding these disorders, but much less useful in understanding the underlying mechanisms. Does that clarify anything? So basically, we can kind of ignore the polygenic risk score in a situation where there's a single strong effect or causal mutation, and that's the case for many of the patients that I'm kind of talking about today.
And the very last question I have is, it's a personal story and question from Soma Halder. Her son is 4 years old and diagnosed with severe ASD. with regression after 2. He's been sent to Stanford Genetics, and he has an MED12L gene mutation. So I'm not sure if you've heard of that one. It's not an inherited one. And then she was wondering if there's any MED12L treatment research being performed at UC Davis.
With 102 and growing genes, there are a lot of families that are in the same situation, where there's more than one kid, but it's so rare still. So I don't know of any efforts on that specific gene, but that's mostly because I don't know what's going on. And I would recommend connecting. I think the Division of Genetic or Genomic Medicine is a great entry point to learn more about what the opportunities are or what's going on at UC Davis.
And I would also just encourage, because I also work on rare developmental disorders with parent advocacy groups, that you could probably type in your gene to Facebook and find more than one parent that has that gene. And that's how it seems so simplistic and silly, but that's how some of these parent advocacy groups got started, and they now have more than one clinical trial going for their subpopulation. So
yeah, the ability to tie together genetic diagnoses across families has really been a game changer in support and in kind of leading the charge to do research on individual genes. So, but again, it's something that you should definitely get in touch with the Division of Genomic Medicine.
How much control is there over the level of upregulation of the expression of the remaining allele in haploinsufficiency diseases? There's no upregulation.
Yeah, I guess there's 2 ways that I can interpret this question. One is just, let's ignore treatment. Our cells can compensate some. So sometimes there's what's called compensation, where the remaining allele actually is turned up without any treatment in order to compensate for the lost copy. Generally, in haploinsufficiency, the complementation is not enough to produce the protein. When we're then, the second idea is, okay, if we're going to go play with turning it up using a treatment approach, how much does it matter how much we turn it up? And that's the question that I kind of alluded to, that we're really interested in, is for some of these genes, some early studies have found that as long as they got it kind of high enough, it didn't matter, and there wasn't a negative impact for going above that threshold that's normally expressed. To me, that's very surprising. And I think that there's a situation where many of these genes, if we turn up that remaining copy too high, it's going to cause many new issues. And we know that in some cases because there are mutations that effectively do that for some of these genes. So I think when we get to the level of, in an animal model or a mouse model, it may not matter that much. But when we get to level of actually clinical and physiological, rescue, that's where this question of how do we more finely control it, I think it's going to really be important.
The UC Davis Mind Institute was founded in 1998 with the promise to reduce and prevent the disabilities that can be associated with autism and other neurodevelopmental conditions. Every day, our clinicians and researchers make progress on that promise. Our groundbreaking research on autism, Fragile X syndrome, chromosome 22q11.2 deletion syndrome, ADHD, and other conditions associated with disability are helping affected individuals achieve their fullest potential. Please visit our website or our social media platforms to find out more about current studies, upcoming events, and how you can help make a difference.